Okay, good evening, ladies and gentlemen, and welcome to this evening's Insights public lecture with Professor Sir Doug Turnbull. I'm Professor Bobby McFarlane. I'm going to be a chair for this evening. There are a few little housekeeping things I have to do before we get started. So there aren't any fire alarms expected, so if they do sound, please panic and run screaming from the building. Walk calmly out of the building and the stewards will direct you in the direction in which you should travel. I've been asked to say a few words about Doug, and he gave me a script which I took down, just far too much, what he wanted to say. He's Professor of Neurology at Newcastle University, and he's been an honorary consultant neurologist at Newcastle upon Tyne Hospitals Trust. Doug spent the last 35 years caring for patients with mitochondrial disease, and he's made truly outstanding contributions to the field of mitochondrial medical research. His focus has always been around research that benefits patients, but he understood from very early in his career that this would rely on a fundamental understanding of the pathological processes involved in mitochondrial disease. And with around 500 peer-reviewed publications, I think we can safely say that Doug's milked that absolutely dry. Doug's research career has garnered him with many awards and accolades, not least of which were his knighthood in the Queen's Birthday Honours List of 2016 and his election to Fellow of the Royal Society last year. However, I think there are two aspects of Doug's career about which he would admit to being very proud of. One is his role in establishing an NHS highly specialised service for mitochondrial diseases that is genuinely an international exemplar for patient care, and the other he's going to talk about right now. So without further ado, please welcome Professor Sir Doug Turnbull.
Thank you very much, Bobby. It's always a pleasure to give a talk at Newcastle University. I realised when I was talking to my wife the other day that I've actually been associated with this university for 49 years. I actually came as a medical student in 1971 and still not got rid of me. So what I'm going to talk about is about mitochondrial donation, but I've actually subtitled this Sharon's Tale. And the reason for doing this is, as Bobby's indicated, that this is very much around our research has always been led by what happens when we see patients in the clinic. So to do something a bit different for these lectures, I've actually invited Sharon Bernardi to come and join me, who started this back a number of years ago, and I'll explain. So if Sharon can just come. So this photo isn't the first time I met Sharon. I first met Sharon back in 1993. Yeah? And this is Sharon with her son, Edward, who I think this is when he was a little bit older, and we'll talk a little bit about Edward's story a bit further on. But at that time, Sharon had, in 1993, had a number of pregnancies, and Edward was the only surviving child. And I think without going through this in great detail, one of the things that happened was, Sharon, that your first two children died very shortly after birth. Yeah. So just describe what happened. It was commented at the time that they weren't, or were poorly at the time.
My first baby was 38 weeks pregnant when she was delivered, and it was normal delivery, 6 hours, and they thought that she'd bled because she was very anemic, and then later they kept saying, mentioned something, acidosis in her blood. They couldn't control acidosis, and that went on throughout the years, and sadly she passed away at 28 hours.
28 hours. Mm-hmm. And then your second pregnancy, you went through exactly the same. You went through the pregnancy.
Yes.
Normal birth.
No, that was emergency C-section. Yeah.
And then exactly the same thing happened again?
Slightly different. He'd already died within the 2 hours with the same problem with the acidosis. As soon as he was delivered, there was a problem with the acidosis in the blood and I couldn't control it. And that affected his breathing and obviously he was put on the ventilator.
Okay. And when we said about the acidosis, they mentioned something called lactic acidosis.
Yes.
So it was a build-up, a very, very high build-up, an uncontrollable build-up of lactic acidosis. Now after that, I think Edward was number 3, wasn't this right?
4.
Number 4. So number 3, a similar problem, is that correct?
Yes, again, a little boy, obviously kept in hospital. Doctors were aware there may be a problem, but they didn't know what kind of problem. He was delivered by cesarean section, and again within less than an hour, again the acidosis was in the blood. They tried bicarbonate soda and everything, and they were going to transfer him to Newcastle, but he was getting worse, and unfortunately he passed away at 30 hours.
30 hours. So again,
So breathing problems.
with the breathing problem. So again, really a situation which was virtually total organ failure. And I think that was one of the things that was needed. Now Edward was number 4.
Yes.
So just give us a little bit of a proper story about Edward.
Edward, after a long stay in hospital again for the one pregnancy, Edward was delivered as an emergency C-section on the Saturday, 35 weeks, and obviously within, I think, again 15 minutes, doctors were prepared that he would have the lactic acidosis. They were more prepared about it. But obviously I just had a epidural, so they did come back and tell me he had come onto a ventilator as precaution, and they had checked his blood. And again, it was lactic. Edward had had a blood transfusion the following day, on the Sunday, and he continued. He was 5'8", but he filled up with a lot of water, and he had a lot of breathing problems. He had the ventilator and was on full oxygen.
Was on full oxygen. But Edward, unlike your other children, survived that neonatal period, but then had medical problems and developed a syndrome called Rees syndrome, which is a particular form of this disease which affects the brain and affects a certain part of the brain called the brainstem. And as you can see, Edward, he'd probably be about 6 or 7 then, do you reckon, Sharon?
I think he might have been a bit younger, yeah.
A bit younger, was already in a wheelchair. And we will come back to Edward's story a bit later. Sharon, this was completely unexplained. We were talking about 27, 25 years ago, and it was completely unexplained, and nobody could really understand how somebody could have so many affected children, because it just doesn't agree with our understanding of genetics. Our understanding of genetics at that time just didn't make sense. I will come back to Edward, but you subsequently had further pregnancies, and your last born child was Caroline. So Caroline was your 7th child. So all the other children died within the first 48 hours, and then the 7th child was Caroline. That was 2000.
Yes.
And a very similar thing happened with Caroline.
Even though everybody, again, was aware, and the blood, Caroline was actually placed on a dialysis to clean the blood out for their lactic acidosis. but unfortunately I think she had an underlying heart problem as well.
So what was noticed during the pregnancy and during the scans during pregnancy is that Caroline had a weak heart. And in fact, Professor McFarlane, his first meeting of anybody with, as a registrar in paediatrics, the first meeting of anybody with mitochondrial disease was looking after Caroline. So Caroline sadly died as well. Edward was somebody that Sharon looked after devotedly, and I think we've got some pictures here of Sharon with her husband Neil. Edward with his Newcastle United shirt on, and Edward was very was an engaging young man. You had a really nice relationship with Edward, and this was probably, would you say he was about 10, 11 at that stage, maybe a bit older? So that sort of age group.
Yeah.
But in Edward's condition, this Lee's condition, is a degenerative condition. And Edward's condition progressed, didn't it?
Yes, yes.
And how old was Edward when he died?
He was 21 in November and he died on the 1st of March afterwards. So
Afterwards. And he actually died on the neurology ward.
No, he died at home.
Died at home. And that was of a progressive condition, which really meant that you were giving 24-hour care to Edward.
With the dystonic spasms.
So he had, and he had some progressive neurodegenerative condition which was associated with terrible spasms which were extremely difficult to control, and Edward became progressively more disabled. It's, as you can see, that Sharon actually has some of her own health problems associated with the problem that I'm going to talk about, but I think the thing that really got us started thinking about these issues and thinking precisely led directly to this research, which is why I'm so grateful Sharon's come along. You can see that this is asking us fundamental questions about somebody that is desperate to have her own children. And yet, what's the explanation that she's lost 7 children due to some form of genetic condition? And that's what I hope to be able to explain to you today. So, Sharon, thank you very much indeed. Thank you very much. So, this is where we start. This is where the story starts, that it was 1993. I knew Sharon over a number of years, and obviously still know Sharon, and she's still looked after by our service. But it really made us think a lot about this problem around mitochondria. So I'm just going to explain, because it was some work that actually Professor McFarlane did, was identified that the problem that Sharon had, and the problem that Sharon's children had, was a problem in the mitochondria. So I'm sorry for those of you that are very scientifically orientated that this is just very basic, but mitochondria are very much the powerhouses of the cell. The main function, they have several functions, but if you think of their main function, it's converting sugars and fats into energy. And what happened with Sharon's children was that they just didn't have enough energy, and it's like anything else, if you can't burn up the sugar, you generate lactic acidosis. This is how mitochondria were formed. And this is obviously a recording from BBC 4, but how mitochondria formed. And they probably formed about 3 billion years ago, when there was a primordial soup of bacteria. And one bacteria got ingested by another bacteria. And the reason why that happened was the oxygen levels were rising. And these mitochondria were very good at using oxygen. They were small, but they were very good at using oxygen and generating energy, and therefore this other cell engulfed them. Very important to realise that, because it helps us explain the genetics later on. So this is a schematic of a modern day mitochondria. When I talk about energy, the energy currents in the cell's ATP, and you can see it being generated there. This is just a representation of a nerve cell, and you can see there's lots of these mitochondria in a nerve cell which are providing energy, and they go throughout the nerve cell. The other thing that I'm just going to point out here, which is so relevant to the story, is that these mitochondria have lost many of the characteristics of being a bacteria, but they still remain some DNA. Mitochondria contain their own DNA, and that's really important for the rest of the story. So what are the symptoms of mitochondrial disease? Well, as you can imagine, if every cell in our body has got mitochondria, and every cell in our body needs energy, pretty well any cell can get affected. As it happens, it tends to be those cells which are heavily dependent upon what we call oxidative metabolism. They use a lot of energy. And those cells tend to be in the brain, in the muscles, in the heart muscle, in the skeletal muscles. You have a lot of muscles in your gut, for example, and that's one of the issues, and that affects many patients. But basically, we see patients with a variety of different symptoms, which has made it very difficult to pick up which patient has mitochondrial disease. It's been a very difficult problem, but something we've got much better handle on now. The other thing you might say is, well, look, I've never heard of mitochondrial disease. Well, mitochondrial disease is a rare disease. It affects about 1 in 5,000 of the population. And the best epidemiological studies to look at this have actually come from the northeast of England. And so it's rare, but amongst genetic conditions, it's fairly common. It's about as common as muscular dystrophy, for example. You just don't hear about it quite so much. And the other thing that Bobby's mentioned, which I think has been something certainly we are very pleased to have set up, is that the poor old NHS gets a heck of a knocking these days, and one of the things is the NHS is a fantastic organisation, and one of the things that we were able to set up was a specific clinical service for patients with mitochondrial disease. And that's one of the beauties of having an NHS. You can have patients from anywhere in the UK and can actually come to one of 3 centres where there are a group of mitochondrial experts. For example, I think there's 5 or 6 consultants that are interested in mitochondrial disease in Newcastle, from Bobby and myself, and Grania and Andy. I mean, there's a real concentrated effort where we actually understand a lot about these patients. And we have a way of seeing patients. We also do Skype consultations. There's all sorts of ways in which we can influence the care of patients. And I don't think this is blowing our own trumpet too much, but I think we've got the best clinical service for mitochondrial patients anywhere in the world because we have this concentration. We see about 1,000 patients a year in Newcastle, so it's a significant number of patients. So yes, we know about mitochondrial disease, and yes, people like myself have been looking after patients with mitochondrial problems for a number of years. But what causes mitochondrial disease? Why do you get it? Well, coming back to this slide, what I did point out was that mitochondria contain their own DNA, their own genes. There are also genes from the nucleus that are important for making mitochondria. These are genetic diseases, so there's either a genetic change in the mitochondrial DNA or the nuclear DNA. I've represented this slightly differently on this slide. So there's 2 contributions from the nucleus and from the mitochondrial DNA. Now, nuclear DNA mutations are important and cause about 80% of childhood mitochondrial disease and about 20% of adult mitochondrial disease. But mitochondrial DNA mutations are very important as well. And as you can see there, the ratio is slightly different, but it is still a very important cause of mitochondrial disease. The thing that I'm going to talk about now, for the rest of this talk, I'm going to talk purely about mitochondrial DNA. So we're forgetting about the nuclear problems, we're talking strictly about mitochondrial DNA. And why is that so important? And why is it so relevant to the story you've just heard from Sharon? Well, the reason why it's relevant is for this reason. Is that mitochondrial DNA is purely passed down from mother to child. There is no transmission, there's no paternal transmission of mitochondrial DNA. So the fact that Sharon has a mitochondrial DNA mutation meant that every one of her children would have that mutation. And that was something that we discovered back in 2000 when Bobby found the mutation. And it's really very important that you realise that mothers who carry mitochondrial DNA mutations will transmit to their offspring. And this is where it started. And this is what we were dealing with back in the early 2000s. And I've given Sharon's story is still the most tragic story that we've come across, but there are many, many families that we look after who have similar stories to Sharon. And what it made us think about very hard at the time was, well, how can we offer something? We know we can't treat these diseases. And this was 20 years ago, and I have to say, we're still no further in treating these conditions. We've still no cure for mitochondrial disease. But what we said to ourselves, well, is there a way in which we can prevent the transmission of these conditions? Because, again, if those of us that have got children, there is a desire for us to have children, and for many people, for some people, there are a variety of options like adoption or oocyte donation. For many of us, we want our own biologically related children. And this is where we came across something called mitochondrial donation. And I'm not going to go through all the basic science, I'm just going to explain what this means. So, I've explained to you that mitochondria contain their own DNA. And it was the mitochondrial DNA that causes Sharon's problems and the problems with all her children. Sharon has nuclear DNA, and all her children had the nuclear DNA from Sharon and from the baby's father. Okay? The characteristics that make me, or Professor McFarland, are basically in your nuclear DNA, but we've got our mother's mitochondrial DNA. The only thing that mitochondrial DNA does is allow our mitochondria to work. It doesn't have any other characteristics. So the proposal with mitochondrial donation is if we take a woman's egg, the egg gets fertilized, and immediately after fertilization you have two pronuclei. You can see one is the female pronuclei and one is the male pronuclei. That's the nuclear DNA that gives us all our characteristics, but the mitochondria are here. Then an oocyte from a donor woman is also fertilized. You've got two pronuclei, one male, one female. You remove those pronuclei, you take these pronuclei, you put them into this egg, you have the pronuclei from both parents, but the mitochondria from the donor woman. So you've got mitochondrial DNA from the donor. Now you're all thinking, whew, that's tricky. Well, it is. But we've got some extremely skilled embryologists in Newcastle working with the IVF team at the Centre for Life, and this next video comes from Mary Herbert, one of our colleagues, and Louise, who is a very skilled embryologist. This actually a oocyte on a Holden pipette. There's a small hole put in the outside of the oocyte. You can see Louise is going in with this pipette to get the pronuclei, which you can actually see down the microscope. It's about 100 microns, so it's not tiny. You can see this down the microscope, and you can see that she's removed both of those pronuclei. And what has to be done is you have to wash those pronuclei in a fluid which is a little bit sticky. And then what happens is Louise then puts them back into this enucleated oocyte, and you can see that they go in. But she's just pushing them in. She tucks them in, and then over the next few hours they will fuse, and that oocyte will develop. And we know from Mary's work that the efficiency for this is about 50–60%, and you can get normal embryos. Normal embryos. Okay. So, and this is where the tale goes, and it's been an interesting tale because clearly the science is tricky, but there is also this component that whilst we published our first paper in 2010 showing this was feasible in human embryos, whilst we could do the science, we weren't allowed to do this. This was against the law. The 1990 Human Fertilization Embryology Act is a brilliant act of Parliament, but it wanted to stop certain IVF techniques, and you need to have a permitted embryo. And this was not a permitted embryo. So, in effect, to move things forward, we had to be thinking about changing the law. Those of you that work with me know I absolutely hate busy slides, and this is exactly the opposite because this is a very busy slide. But it just shows some of the pathway that we had to go through. That we were even thinking about this back in 2001, because we were discussing this with the chief medical officer. But in 2010, there was a hive of activity because we went to the Department of Health and said, look, we think this might work. Do you think we can start to change the law? And this raised a huge number of issues. And I'm not going to go through them all, but I think there'll be people in this room that think, well, that's a great idea, and other people think that's a bad idea. So there's huge ethical issues. If you're doing something like this, you've got to think about, well, it's not just, of course, Sharon and patients like Sharon or Professor McFarlane and myself, of course we think it's a good idea, but what does the general public think? Is this something that the public will think acceptable? Because politicians listen to people. So there had to be a large public consultation. And all of this was going on, and it was a quite intense time over a period of 5 years, where there was a number of things that were going on to try and see whether or not this can come to Parliament. And there was a whole group of us that were working, both engaging with different groups, doing the science, and thinking about the policy. Because how do you change the law? How many of us have changed the law? How many of us have ever been to Parliament? How many of you met hundreds of politicians? So it was a really important stage. But what I want to stress is that the most important people in all this were the patients. Because there was no way that this would have gone forward if the patients hadn't been advocates for this. And this is a really important lesson. And this is just some examples. For example, we've got Sharon and another lady with a very poorly child who was very much involved in newspaper interviews. Newspapers TV programmes don't want to talk to boring people like me. They want to hear the story from the patients. And it was very important that the patients felt that they wanted to get involved. We used to go down to Parliament. We used to go and see politicians and say, look, this is why we think it's important. Please, please, will you support it? This, and I think we probably only got to Parliament as well, and is because at the time, the Prime Minister at the time was David Cameron. And of course, David Cameron had lost a child to a genetic condition, and I think that gave us a certain sympathy. Those of us that were actually there in Parliament, there was a parliamentary debate, and I don't know if people know that the Speaker always calls up people who've got different opinions, so you have no idea which way it's going. But the parliamentary majority was significant, was 382 to 128. It was a massive mark for the majority. Then a month later it went to the House of Lords, and the percentage majority was even greater. So it was a really important step that this was a law that was changed basically because patients needed this to be able to move forward. It was signed into law in 2015. I'm just going to remind people about that date, 2015, because you're going to think, well, what's happened since then? I'm going to try and explain that a bit later. But I thought, one of the things that we've worked with, and Sharon will know this, that we work very closely with a charity, and we work very closely with a charity called the Lilly Foundation, which was set up by a mother that had lost a child to mitochondrial disease. And that charity worked tirelessly to help, and then they put together a video, which I'm going to show now, of just how intense that process was back then.
The United Kingdom is said to become the first country in the world to introduce legislation allowing the creation of human embryos from the DNA of 3 people.
This is a bold step for Parliament to take, but it is a considered and informed step.
The first babies with 3 genetic parents could be born in Britain as soon as next year.
It offers hope to families affected by a wide range of diseases caused by faulty genes passed from mother to child.
Describing mitochondrial donation as producing GM babies, or indeed 3-parent babies, are phrases that are wildly misleading.
It's not fair that if there's a scientific technique out there that could resolve this happening and stop this happening in the future, that we're not using it.
We're sitting there with a technique that could potentially prevent the transmission of these diseases.
Mitochondrial disease is an inherited disorder that affects around 1 in every 6,000 babies.
Could help about 150 couples each year who are at risk of passing on certain genetic diseases.
It's aimed at stopping infants from being born with inherited diseases. The new method would take genetic material from a mother, a father, and from a donor woman with the aim of preventing deadly diseases being passed on to the child.
It would just be incredible. It is like a light at the end of the tunnel for them.
Her children would also inherit the disease unless MPs vote to allow a controversial technique to change the genetic material she passes on.
Mitochondrial disease is just horrific. The way that it affects children, and it shortens lives, and the suffering that children and adults have to go through.
I think this is an important advancement in medicine to allow women to have healthy children.
I think we have to be really clear that there is currently no cure and no treatment for mitochondrial disorders. It's debilitating, it's cruel, and families and parents have to watch their children die.
The eyes to the right, 382. The nose to the left, 128.
Right, so there we are, 2015. And I suppose there was a little bit of us that said, we've done it. You know, we've done the science, we've got to Parliament. And then it kind of dawned to us that actually some of the biggest challenges lay ahead. So how much do you think mitochondrial donation costs? I'm going to explain a little bit about the pathway, but it probably involves 2 or 3 rounds of IVF for the woman with mitochondrial disease and IVF for the donor. So you can see that this is a significant investment. So actually, getting the funding to do it is really important. But also, we're talking about a totally new IVF technique. You've got to make sure that what we're putting in place is absolutely right for the patients. And an awful lot of work from my colleagues has put this in place. And I just want to kind of go through and explain. So the first is, I've explained to you in the kind of 35, 40 minutes that we've been going about mitochondria and mitochondrial DNA. But actually, it's hugely challenging for people to really understand and make informed choice. Because one of the things that's so important here is that patients should make informed choice. It's not what we think, it's what the patients think. And really one of the first key things is this reproductive advice clinic, which is run by my colleague Ronnie Gorman and Catherine Freeman. And this is looking very carefully at the genetic factors, the families, and of course, women that have these mutations actually have disease as well, and discussing about their ability to get through pregnancy. So it's not it's not a quick 5-minute visit to the doctors. This is a substantial amount of work, and there's all sorts of videos to explain the way in which the process works. If patients decide that they want to go down one of the IVF routes, and I'm not going to go into any detail about the various options, but then go to a separate clinic which is run by Bobby McFarland and Jane Stewart at the Newcastle Fertility Centre, which again, Bobby goes back over some of the mitochondrial side, but Jane introduces all about IVF. And there's a great tendency to think, oh, IVF's just straightforward. IVF is not straightforward. And of course, there are a lot of issues. One of the issues, for example, is if you're a bit older, you simply do not produce as many oocytes. And of course, quite a lot of people have been waiting for this and are in the older age group, certainly in terms of the reproductive life. The next point I want to make is that, of course, for this to work, we need to have donors. So, many people will be blood donors. Have you ever thought about being an oocyte donor? It's not quite as straightforward because you have to go through, you have to volunteer, you have to be assessed, you've got to be assessed, and you've got to go through the IVF, where you're collecting fresh eggs. Because this is done with fresh eggs from the donor and frozen eggs from the patient. So it's a remarkable act of generosity from the donors. We're always looking for donors, and especially people who live close to Newcastle. They've got to be women of a certain age and a certain level of healthiness, because clearly there is a need to make sure that when you're donating eggs, that you're giving there's plenty of eggs there to be used for this technique. So again, it's something that we are working on to try and increase the donor pool. As patients become pregnant, we need to make sure that we monitor pregnancy. And that's something that Catherine Freeman does throughout the pregnancy and keeps in touch. And then, again, this is a very busy slide, but it's really just important to see that there are several steps that Bobby's devised to look after any children born. And the ones in light green are actually what happens already in the NHS, and those of you who've either had children or grandchildren will recognise that there's many steps that the NHS already do. And they are fantastically good at monitoring. We just need to make sure that we collect all that information. But they are all going to come at an 18-month review to be seen by Professor McFarland, and then at 4 or 5 years. Obviously, the idea here is to actually try and follow up these children for many, many years. But it's very difficult for us to put a pathway in place. So, how do we fund it? And I think this was something that you would have thought would be incredibly difficult to do, but actually proved to be very easy to do because of the people that we work with. So this is an IVF treatment, and there wasn't a mechanism for NHS England to pay for it, because they will only pay for proven treatments. But what they would pay for is what happened was with our colleagues at the Wellcome Trust that fund a lot of this research. What happened there was that they said, well, they'll fund Professor McFarlane to follow up these children at 18 months, but the NHS England said that they'd pay all the excess treatment costs. So there is now funding available for the first 75 children born, paid for by a combination of the Wellcome Trust and NHS England. So where are we? It has taken a significant period of time to get this up and running. We were not able to do this, despite the law change, we weren't really able to do this till 2018. And the reason for that is there were a number of things that had to be done associated with the license. There are 20 licenses for individual women that have been granted. I cannot say anything more about that because obviously that is confidential information. But I think equally important is that there's been well over 150 women that have been able to come to our service and actually have major advice, reproductive advice, giving them the best possible options available. And one of the things that we're doing is actually seeing women earlier and earlier, younger and younger, so that they are young in their reproductive life that they're making these decisions. So I'd like to end by saying that it's always, when people get up and talk about research or talk about progress, of course, it's always a teamwork. I've been very fortunate in Newcastle to work with great colleagues, and some of those colleagues are up here, Bobby and Grania are in the audience, and Lindsay. And it's been a great privilege. And I think probably the most important people in all this research are the patients. So thank you to Sharon. Thank you very much.
Thank you very much, Doug. We'd like to take some questions from the floor. Got one there. I'm sure we've got a microphone.
Thanks. Will the children who are, any children who are born as a successful result of mitochondrial DNA donation, will they be carriers of the maternal DNA defect?
Right. So any male children born will, of course, not transmit on anything that's carried over. It really depends how successful the technique is. So in theory, we could potentially completely change the mitochondrial DNA completely. Whether or not that is going to work in practice is something that we're looking at now. But in theory, if this is 100% successful, then none of the mother's mitochondrial DNA will be in that.
Very much. That was absolutely fascinating. In terms of screening for mitochondrial disease, how do you go about that? Do women come forward, or how do you find out that they've got a problem to begin with?
Yes. So a lot of the patients that we see are actually patients from families that we know have got mitochondrial DNA disease. That's not 100% of the time, but it's a lot of our patients. So we actually see quite a lot of families, and a lot of the patients that are being referred to Grania and Bobby at the moment of patients who have been seen out around the country because they have either family members or have a characteristic clinical syndrome that means that they're highly likely to have a mitochondrial DNA mutation.
Hi, I found it all very moving. How did you know? Did you know before that you had a mitochondrial problem, Sharon, or did you find out because of the births?
It was because of the births. Sharon was very much at the forefront of our diagnosis. We did not know that Sharon had a mutation in her mitochondrial DNA until after the birth of her last child. Because it was very difficult in those days to find a mutation. And that was when you knew something was amiss because of Edward, didn't you?
I did it better because my mum had also lost babies, but then that was in the 50s.
Yes.
They died and they didn't have an answer.
Yes. So Sharon's mother also lost.
Cousins as well.
Yeah. So it's certainly something in the family that, with hindsight, of course, we could work that out. But you know, we are talking 20, 25 years ago, and DNA technology has changed out of all proportion now.
I spent roughly 3/4 of my career in healthcare in the USA. I wonder if I could trouble you to give a sort of global overview. I guess this whole thing will go down too well in the States because of their antipathy to stem cell activities. But again, what are things like elsewhere in Europe, like Germany and Switzerland?
Okay. Yeah, I think that's a very interesting question. We in the UK are very fortunate to have the HFEA Act, and we also have a very pluralistic society which deals with many of these issues. In the United States, there is no regulation of IVF at all. And to describe it like the Wild, some of my American colleagues describe it like the Wild West. However, there is a complete restriction on doing things like mitochondrial donation. They tried to do it in the States, but certainly with the current president, there's no way it's going to move forward. It's something that I'm actually involved in a Royal Society Commission at the moment, just looking at different forms of IVF techniques and newer forms of trying to prevent transmission of nuclear genetic diseases, and it's very difficult in the United States. So the States, it's an absolute no-go. Other countries around the world, so this has been done in Mexico by, I can't remember, it's called ethics something. You go to a country that doesn't have an ethics committee, and you go and do it. There are countries like Ukraine, where they're doing it, because there is no regulation of any IVF or any ethical. So it's a real mix. We are getting a lot of queries from Australia and from Europe to actually set up into this year, and that's one of the things that Professor McFarlane and I do, while they try and change the laws, certainly in Australia. In Japan, there are visitors from Japan trying to set this up. It's interesting across Europe, there are very, very different views. And for example, in Germany, you're not allowed to do any embryo research at all. So it's very different in different countries. Having never worked in the IVF area, I can say my eyes have been opened wide by very different practices.
Question here. Look, I'll come back to you.
I don't really need the mic, I don't think. Excuse my ignorance on these matters, but is there a theoretical possibility with refined screening and widespread use of these techniques that you could actually look to eliminate the genetic passing on of these, or the passing on of this?
Right. Yes. So that was a question about whether or not you could eliminate this disease. I think it's unlikely. I think what we'll be able to do, because you're always going to get new mutations, and that's one of the things that we're always. There's also some restrictions on the use of this particular technique, which means that certain forms of milder mutations are not going to be suitable. And there are some complications here, which I won't go into, that if you only have slight change in your mitochondrial DNA, then there are other techniques that would. The other thing is that it's very much patient choice as well. You know, we got very much accused when we were having some of these discussions that we were trying to force patients to make decisions. There'll be people who will be making a very different decision to other people, and we know that from some of the clinics that are being held. I think this will represent a technique that is used for a percentage of women that carry these mutations, but it won't be suitable for all.
Is there a pre-screening possibility for women rather than going through the process of giving birth and then having to see?
Yes. So I think we've got a very effective way of picking up most women that carry mitochondrial DNA mutations through family screening. And we are very, very active at doing that. There is, one of the things that's changed completely, probably more than anything else over the last 10 years, has been the introduction of genetic testing and more genetic testing. So will we ever get to a stage where all couples go through a whole genome sequence to look for rare genetic diseases and then try and select down different ways to prevent that? I think that it may well come to that at some point. It's not there yet, and I'm not sure everybody would want that, but there would be a way in which we could pick up many of these diseases by just sequencing everybody. But is that really what we want? And I think that's another discussion that is going on. But of course, you pick up all sorts of things. 1 in 64 of us will carry a cystic fibrosis mutation. What about a BRCA2 mutation that causes breast cancer? You know, it opens up a whole number of things and a whole dilemma that I think society will have to look at, but we're not quite in that yet.
So a question just at the back.
Well, what is the explanation for the very different outcomes for a patient with abnormal mitochondrial DNA who enjoys an apparently normal and healthy childhood and adulthood, and her children with the same mitochondrial DNA who die at birth?
Yes, I think, again, a really interesting question. What we found out is that mitochondria don't work in isolation, and your nuclear background will clearly make a difference. It's a very difficult experiment, very difficult to do that when you have what is a rare mutation like Sharon's, but we've some patients where we've got about 350 patients who've got the same genetic mutation. And it's quite clear that the nuclear genetic background is affecting the phenotype. So that is the explanation. What that nuclear factor is, we don't know yet. But there is one of the people in our centre is looking for that, because if we could identify that and what that gene does, then it might introduce a treatment for mitochondrial disease. So it's a very important thing. But it's a very difficult thing to look for within relatively small groups. Looking for something in looking for variation in 300 people is still very difficult from a nuclear genetic perspective. If you looked at 3,000 or 300,000, it would be much easier. So I'm just saying it's something that we're looking at, but it's proven to be a challenge.
Any more questions? Second parliamentary, one.
Another question. Do you think it might be possible in the future to harvest healthy mitochondria from cells other than liver?
I think it's unlikely. People have asked this before as well. I think it's unlikely. Mitochondria in oocytes have a particular characteristic. And they, I mean, if you look at them down the microscope, they look very different from mitochondria in heart cells or muscle cells. And I think if you start putting the wrong mitochondria into an oocyte, I think it would have an adverse effect on the oocyte. So I don't think that's going to be, because people have often said, well, why can't you use sperm mitochondria? Well, you simply can't. It's just too different.
Any question here?
Yes.
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 Thank you.
Got my eye on her. Yeah. On my radar.
Is this, are there links developing with the mitochondrial problems and sort of some of the less understood conditions that affect all those similar organ systems like ME and things like that? Is there much?
Yeah.
Sorry, it's really vague, sorry.
Yeah, no. I think it's an area which we get asked about a fair bit, and there is a lot of similarity with some of the symptoms that we see in our patients. With fatigue is a prominent feature. Understandably, I mean, I didn't put that on because it's difficult to show a picture of fatigue. So people do worry whether or not other fatigue syndromes are but there is no evidence that things like chronic fatigue syndrome are actually due to a mitochondrial defect. There are some slightly scandalous tests being done by certain people for a certain amount of money, claiming that they can do it, but there is no evidence, as far as I'm aware, that there is a substantial mitochondrial problem in patients with chronic fatigue. The other thing that happens is if you have fatigue and you don't do much exercise, you get less mitochondria max, as much as it would happen if I start doing exercise. I get less mitochondria as well.
The lady up there in the white top, yeah.
First of all, I'd like to say what a fascinating talk you've given. Secondly, I'd like to say that having lost one child to a genetic disorder, I cannot imagine what Sharon has been through. You mentioned that 20 licenses have been given to couples with this to try and eliminate this condition. Have you actually had any pregnancies or any live births yet?
I can't comment. It's very difficult. Again, we try and work very closely and we try and I believe the scientific process should be open and shared. But of course, once we start coming to people's personal medical histories, and we're just simply not allowed to comment.
We're all done. We're all done. We're all done.
We're all done.
Okay.